My name is Dave Caron. I'm a professor at the University of Southern Cal. I also have a small consulting company, Blue Water Science. Purpose of that really is to use extant scientific knowledge to try and help water managers who often face issues where they don't know what a HAB is, they don't know what caused it, they don't know how to mitigate it or manage it, and you'd be amazed at how large that knowledge gap is sometimes. And so this is just one way of kind of giving back to the community. I know academics, we ask a question, we do science to answer that question, to ask 5 more, and we keep going. And there needs to be a connection between the knowledge we have, extant knowledge we have, and those water managers who need it. So I'm just going to get into it here. As I said, no complicated figures. I'm not going to belabor this. We've heard a lot of the talks that were already given in this room this afternoon were really good in kind of the lead up, and I'm going to ping on a number of them. And I'm not going to cover a lot of them or any of them in real detail because a lot of that's been done. But I'm going to, I hope, fill in a few gaps and maybe put in a couple of few caveats. But the only saving grace that the cyanos in Clear Lake have is they're beautiful at different levels. My technician calls the one in the upper left the Picasso of Clear Lake. It kind of looks like that. Okay, I lied. One complicated slide. There are a lot of drivers for blooms, and we're going back to what drives these blooms, what makes them spin up. There are a number of extrinsic factors that we've paid a lot of attention to. We still do. The answer is in there somewhere. Temperature or climate, and I kind of have it quoted there because we hear a lot about that. I'm going to talk about it in a second. But drought, fire, nutrient loading, all of those things are getting quite a bit of attention. All of them undoubtedly play some role, but not necessarily how we might think. And the example that I'm going to give here is we published a review paper last year. It's Jamie Smith, who's at Squirt now. She was a student in my lab. We started this. Sarah's on this one. We went back, as review papers do. We dredged up data out of the literature. What you're looking at very simply is temperature. The annual cycle of temperature in Clear Lake, there's no big surprise there. There's a max somewhere here in the summer. But you'll note that there are sort of chunks of data here. What we did is we took data that we could get from about 1960-ish all the way up to 2020. And we plotted each month according to the year that it was collected. And there are a couple of things that you can take away from this. So we heard earlier, I think it was Michelle, who said that there is definitely a rise in air temperature over the past several years in the Clear Lake area. That's true. But actually the maximum temperature attained or unattained in the system has really not changed very much from 1960 to now. So the lake gets as warm as it gets, but it doesn't get warmer because there's warmer air temperature out there. So that was kind of striking. The average temperature doesn't seem to change too much from year to year either. These little blue lines are the regressions of the data within a month for all of those years. And the flat slopes there on those indicate that they're just kind of flat from 1960-ish on. However, there are positive slopes here, you can see for some of these months, and that indicates that from 1960-ish to 2020, it's gotten warmer in each of those months as the years have progressed towards the model. So we're actually seeing a warmer temperature in the lake earlier in the year, and that I think is a major way that the temperature is playing out in this system. There are also at least one, I think it was October, that had a negative slope. So it may even be healing over a little quicker. That is, it's getting cooler quicker as well, but not well into the fall. Well, this means that the growing season for the cyanobacteria, they love it warm. We know that is probably longer because of that increase of temperature earlier in the year as we progress towards the present. So it's an interesting twist. And I think it's worth noting, it's just worth kind of keeping in the back of our heads, because even if we're not seeing a real strong relationship between time and lake maximal temperature, if there's a longer growing season, we're letting those cyanobacteria do their thing a little bit longer. All right, I'm not going to belabor this. There's been a lot of talk about nutrients. Of course, we always focus on nitrogen and phosphorus because of all the elements that things need to make themselves Nitrogen and phosphorus in aquatic ecosystems are usually in the shortest supply. And these elements are different in their activities, and we've heard a little bit about that today. I'm sure you all know about it. Nitrogen is what I would call fungible. In fact, there's tons of nitrogen in the air, but it's present as molecular nitrogen, N2, which almost nothing can use except for bacteria, including cyanobacteria, and they can take molecular nitrogen, of course, and do what we call nitrogen fixation and break those atoms apart and use the nitrogen atoms in biology. They can also go the other way. They can also denitrify or take organic nitrogen or inorganic nitrogen, a single element or single atom, and make it back into molecular nitrogen. The bottom line is when they run out of nitrogen in a system, well, what cyanobacteria just come in that have the heterocysts that Sarah just referred to, they fix nitrogen, they make their own. Phosphorus, though, is immutable. Phosphorus atom is a phosphorus atom. It's hard to get rid of unless you lock it up with something and send it to the bottom forever. But you can't, there is no atmospheric source of nitrogen to speak of. So it's typically what we use to attack the system. And attack meaning, what do we limit? What do we get out of the system in order to prevent blooms from taking place? And of course, phosphorus is the thing that there are most targets, at least in freshwater. Marine systems are a little different. They tend to be more nitrogen limited. But we wanted to look at, so in 2007, TMDLs for phosphorus were enacted in Clear Lake. Have they made a difference? Well, again, we went back into this data set, got the numbers that we could get. It's unfortunately a hiatus in here. But if you look at these later dates from about here on, has it made a big difference? Not so much. And I think this was also reiterated in one of the talks, I think it was in Angela's, where actually phosphorus TP, total phosphorus, looks like it's going up in the last number of years. Was it 1985? Is that the bad news? But it's going up. So the TMDLs are not having the effect that we want them to have. And so, that's kind of where we are. And unfortunately, that's confirmed by documentation of massive blooms. And we just heard Sarah talk about some of those data. Another way of looking at that, this is another figure from J.D. Smith's paper, and I give her full credit because the probability analysis there was over my head. But what she did is she looked at the probabilities of phosphorus concentrations with that huge data set of all those numbers. And just to make that a little clearer, if you look at the green boxes here, we compared what do you get before the TMDLs, or what do you expect to see, the highest probability, what kinds of phosphorus concentrations relative to after the TMDLs. And the blue line, bluish line there, is before the TMDLs. And what that high peak above the reddish peak is telling us is that there's a pretty high probability of seeing low phosphorus concentrations prior to the TMDLs relative to after the TMDLs were enacted. But on the flip side, there's a slightly higher possibility, maybe the same, maybe a little higher, of seeing high concentrations of phosphorus after the TMDLs relative to the time before those TMDLs were enacted, the blue line. So, and this is just another derivation of the same data. So in essence, what we're seeing is there's really not a lower probability just because those TMDLs were enacted. And what we did to compare data sets here, we just took the total phosphorus for 10 years before the TMDLs, then we compared it to 10 years after, because we had just about that. And there's that big gap, if you remember, in the middle of phosphorus. So we couldn't do everything against everything, which we would like to do. Okay, so what does this mean? It means that there are huge loads of phosphorus that are already present in Clear Lake. This is something we sort of know. And the corollary to that is that the key to understanding the drivers of these massive blooms is in large part dependent on understanding how these internal loads of nutrients in the lake are made available to the phytoplankton community. The less fun way of saying that is the TMDLs don't matter. It's coming into the lake matters a heck of a lot less than what's already in the lake. We could have blooms from now until doomsday if we didn't do something about those internal loads. And that ultimately is, I think, what we need to do in order to be able to address the issue. Again, I don't want to belabor this, but because it's been sort of covered nicely. Alicia mentioned some of this stuff, which was really nice. And essentially, this is from a very different paper, but it shows the phosphorus sediment mantra, which is you have phosphorus in the sediments because anything growing and dying in surface waters will sink down eventually to the sediment. It is normally decomposed up in the water column. And as you go from mesotrophic to eutrophic or hypereutrophic systems, and Claire is definitely on the end of the hypereutrophic, you have quite a bit of burial of phosphorus in the sediment. But if you're using the oxygen in those lower waters, you're creating hypoxic and anoxic situations. And that is actually chemically, that is releasing phosphorus from the sediment and it goes up into the water column. And this is what many of the approaches try to fight. There's a low oxygen in the water, and we've heard a number of talks that have talked about the DO concentrations in the lake. This doesn't mean that if the system were completely aerobic, that you're not going to get phosphorus release from the sediment, because there's a ton of microbial degradation, decomposition that's taking place. And if you pump oxygen in down there, it is going to be done aerobically, and that process will decompose and release a lot of phosphorus. But it is presumably quite a bit less than if you have hypoxic or anoxic conditions. Okay. So oxygen is a key factor mobilizing phosphorus. We kind of know that as well. As part of a study we did in 2019 and 2021, and it was kind of a number of things. Sarah was involved in it again. Part of the data that went into the review paper came in the study. But some of the study that we did was molecular in nature, and we actually now have a huge molecular data set to look at not just the cyanobacteria, but the other bacteria and the eukaryotic organisms in the water as well. That's just coming out now. But if we go back to the studies that we did with remote sensing, we've heard a little bit about this again, Malaysia. We used along those purple lines, we had the Xylem people up and they flew their eco mapper through the water. And this is an instrument that goes up and down as it goes forward. There are instruments up in the cone, the nose cone, that measure things, and then you can come out with beautiful two-dimensional plots of what everything looks like throughout the entire lake. It's a great whole lake spatial variability kind of. The other instrument we used, we borrowed from oceanography. There's a great instrument called a wire walker, and it has a big float at the surface. It has a wire that goes down, and it has an instrument on it that's basically a mule. It carries whatever you want it to carry. The system ratchets down the water column based on wave action at the surface. So as long as the waves are moving, that thing is ratcheting slowly down, taking measurements from whatever instruments you put on it, all the way till it stops. There's a trigger at the bottom, floats up, and then it ratchets down again. So you get every several minutes or so, depending on the wave action, you get a vertical profile of all of those sensor measurements just continues. And we were able to run that for about a month in 2020. And I'll get back to that. But just stopping with the eco mapper for a second, I mentioned you can get these beautiful whole lake kind of profiles. And what you're looking at here is information from upper arm, Oak's arm, and lower arm. And the top one is temperature, then dissolved oxygen, chlorophyll, and pH. And you don't need to know or take in any of the specific numbers here, but it's just that gestalt. So if you were wondering, is there more chlorophyll in Oak's arm than in lower arm than in upper arm? Well, yeah, and you can see that with your eyes pretty much. But it gives you an indication of some of the variability that's in there. And these are on the same scale. So, pretty much upper arm almost looks oligotrophic here, but it's not. And then lower arm is richer than that. And then the Oak's arm in this particular year, 2020, was extremely rich. Dissolved oxygen is sort of a different story, but then you get these beautiful gestalts. Okay, so that's kind of a nice piece of information. It doesn't put us hugely farther along as to how those internal nutrient loads are becoming available, but it does give you a nice picture where you compare all three of those arms, which is tough to do because it's a big lake and it's hard to get around. The wire walker, on the other hand, we put it pretty much at the narrows. We put it there because it's a rough area in terms of wind action. And remember, this is a very wind-driven lake. What you're looking at in the top panel here is wind direction and velocity. And then that is all averaged out in this little pie diagram over here. And what that is showing you is that the wind direction is predominantly from the northwest to the southeast. We know that. This is the temperature that we get. Remember, this instrument is going up and down. And if I can draw your attention to this one, we're running out of time. This shows you the depth of the instrument. So it's just the depth sensor. And it's showing, by showing these like tons and tons and tons of vertical lines, it's showing every vertical profile that the instrument is making. That's neat. That's cool. But there are interesting little gaps here. And this is where the story is going. If we take a chunk of that and we expand it, so this is about a 3-day span in the middle of August, where we looked at those vertical profiles and we looked at where these gaps were, and we looked at what was happening where those gaps are. So what's going on is that the instrument is going up and down until the wind stops. And then the instrument pauses wherever it is. And then as soon as the wind picks up again, so the waves are big enough to move the instrument, it keeps going up and down again. And you'll notice that these bluish, which are low DO values, are often after these pauses. So if we take 1 day of that and expand it, this is the same data, but we've blown it out tremendously. You've got the instrument going up and down. The light blue line again shows it's going up, going down, going up, going down, going up, going down. And then it has a series of minutes to an hour or so where it pauses, goes down a little more, and then it pauses quite a long time, and then it goes down again. The wind is going, the wind is stopping, the wind is stopped for that period of time. If you look at that top right blue line, that's dissolved oxygen. And when the instrument goes down to the bottom after it's done its little pause at the top, it's just deadpan, right? It's just, the oxygen's just going where? And we're not within that 2 meter range of the bottom that I think Alicia referred to as the most toxic in the lake. It's a little bit above that because we need that wire in order to get the instrument to pop back up. So we have a system where when we have even a short pause in the movement of the water because the wind has died, we have a drop, significant drop in dissolved oxygen. That, of course, presumably means a drop in the phosphorus that's available. So this, a rise in the phosphorus that's available. So hypoxic and anoxic events, they set up extremely rapidly. So like there have been publications on the diel periodicity of this, but this is super fast. It means, for one thing, that the redox undoubtedly plays a role, but it points to a very, very high oxygen demand in those sediments. There's tons of organic material and bacteria just ready to go. And they create that anoxia very quickly. So we have these high resolution temporal, what we call pulses of phosphorus during these short anoxic or hypoxic events, and they could be very important in the way the system works. They could stimulate cyanobacterial or algal blooms. They could prolong blooms. You've got a bloom that's going, running out of nutrients, you get one of those pulses. Once the wind returns, that phosphorus that is in the deeper water gets mixed up on the surface. Or you could induce changes in dominant taxa. We still don't understand why one cyanobacterial bloom turns into another, other than the classic explanation of cyanobacterial nitrogen fixers will come in and dominate the system if there's a lack of nitrogen, but then they give way to other organisms that are not necessarily fixing nitrogen. But those changes in dominance could be really important. The process is simple, but the rapidity of the shifts between the oxygen and hypoxic or anoxic conditions, I think it's pretty remarkable. And it's kind of an important aspect we need to think about for these winds. And I think that's it.